All right, so here we're going to talk about regulation, and we're going to start by talking just in general about regulation as well as regulation of prokaryotes. Now, the objectives are basically to know the purpose of regulation and how, in general, prokaryotic gene expression is going to be controlled. So prokaryotes are going to use a mechanism that's called an operon. Now, one thing to remember with respect to protein regulation is that genes are going to be regulated at the level of the promoter. And so you're going to have some sort of initiation event that's going to require some gene to be made. Now, some genes are going to be made constitutively, and so these are going to be our housekeeping genes. So these are genes that cells need in order to survive. So these are typically always on. And these can be regulated, and the regulation is to turn them off. Now, other genes that are not constitutive, are not housekeeping, are going to be regulated at a promoter sequence, and it's going to be regulated by turning them on. So some signal is going to have to be made for these other genes or non-housekeeping genes to be turned on. Now, the operon is a cluster for prokaryotes, and these are going to typically have genes that are going to be coordinated together. So genes are going to be transcribed together, and they usually have a similar function. Now, some of these are going to include structural genes, and some can include regulatory genes, or regulators. And so typically what ends up happening is that you are going to have an operator site, and this operator site is going to be bound by a repressor. And this repressor turns off the gene. And then you're going to have some sort of inducer. An inducer is going to break the bond between the repressor and the operator. So it's going to release that turn off, that break. And at the same time you have an inducer for prokaryotes, you're also going to have some activator. So it's going to bind to a sequence that's going to promote the transcription of the genes associated with this operon. Now, with respect to our prokaryotes, you're going to have some regulatory binding motifs. These are going to be a helix turn helix and a zinc finger. And a helix turn helix is a 20 amino acid alpha helical segments that are separated by a beta turn. So you have 20 amino acid alpha helixes, and these are going to be separated by a beta turn. And then these are not very stable, and usually one of the alpha helixes is going to be the recognition sequence that is doing the regulatory part. And then we have our zinc fingers, and our zinc fingers are going to be 30 amino acids, and they have a long loop that is held together by zinc. And zinc does not interact with DNA. Now, the zinc finger is also going to be in eukaryotes, and the the mechanism with which the zinc fingers are going to bind to the DNA differs from one protein to the next. And so we're going to have different zinc finger motifs as shown here, in which you have the zinc that is going to hold together these loops of the amino acids. Now for the prokaryotes, just basically you need to understand that the genes are going to be regulated. The regulation is going to be at the level of transcription by regulatory proteins. You can have proteins that are going to suppress the regulation. So for housekeeping genes, for example, they're always going to be on, so you have to have a negative regulation. Or you can activate transcription. So this is something that the cell doesn't always have on, but it needs, and so this is going to be positive regulation. In bacteria, we're going to have our genes on an operon. Typically, genes linked together have similar functions. You can have your transcription of non-housekeeping genes blocked by repressors, and the repressors are going to be relieved of their blocking by those inducers. And then regulatory proteins are going to be two main types in our bacteria, our helix-turn helix structures, as well as our zinc finger domains.